She's a world leader in bioinformatics, an interdisciplinary field that develops methods and software tools for understanding biological data. She led a team that used DNA sequencing to determine that the Ebola virus was spreading from human to human, not from mosquitoes or other animals. She was named by Time magazine as one of the 100 most influential people, and also a Person of the Year for her work as an Ebola fighter. She is the recipient of Smithsonian Magazine's American Ingenuity Award. Delegates, please give a huge hero's welcome to Dr. Pardis Sabeti.
Thank you. That is some amazing energy. Well, welcome, and I'm really excited to be here with you guys tonight. I'm going to tell you a little bit about my work. I'm an MD Ph.D. who uses genomics to try to understand disease. And the work I do is really, in the last few years, has been thinking about how to transform the way we respond to infectious disease outbreaks using genomic information. So you all will likely know about the story that sort of really took fold in March of 2014 when Ebola was first detected in Guinea. And you were probably all engaged and involved as the first cases came into the United States and learned a lot about it. I'm going to tell you that story again, but I'm going to give you a few facts that you might not know. And one of the reasons why I know a number of these facts quite closely is because of the way that my team became involved during the outbreak. So we had been working for a decade in Sierra Leone at the Kenema Government Hospital. And this is folks from my lab joining Dr. Humar Khan in the blue shirt and his team in Sierra Leone, bringing diagnostic tests for Ebola when that outbreak was first declared. So Guinea is very close to Sierra Leone, and it shares a border with Sierra Leone and Liberia. And so we recognize that if the Ebola outbreak was to spread, it might come into Sierra Leone, and we may see it at the hospital. So we made sure that the hospital had basic diagnostic tests to pick it up. And as you may know, as individuals from around the world tried to understand how this outbreak had first started, they believe that it had started by a single individual, an 18-month-old boy named Emmanuel in Guinea, who was playing in a tree that had bats, and it was disturbed, and he fell ill. And then soon after, his family became ill, loved ones, caregivers, and the virus began to spread. But by the time, so that likely happened, they traced that back to sometime in late of 2013. But by the time the outbreak was actually declared in March of 2014, it had already gone to all of these different districts of Guinea. So it's already spread, and around 60 people were picked up at that point by the time they understood that there was an outbreak going on and that outbreak was of Ebola. And then soon after, in April, we started to see cases in Liberia, and you can see how it's moving through that entire region. And then by the time it hit Sierra Leone, it came in May, and it blew up. Right, and that's when it suddenly took over and everyone became really concerned. And suddenly it almost was in every district in those three countries. And this is another way of looking at what was going on. So you see the number of cases over the months that happened. And at first, the cases were increasing, right? When it was in Guinea and Liberia, it was increasing quickly. And it was already the largest outbreak of Ebola that we'd ever seen and documented in modern times or in history. But when it had hit Sierra Leone, this is how the trajectory changed. It suddenly hit an inflection point, and it was blowing up. And individuals were trying to figure out what happened. And part of it they thought was that, because the WHO and MSF and the other organizations were overwhelmed, it might have just, gotten out of their control, or that it hit bigger cities, and that was what was going on. But there was still a question mark. Why did cases suddenly start to emerge? And there was a clue for us when we started looking at the sequences of the virus. So my team had been working in the country for this long period of time. We'd already set up capacity to do both testing, diagnostic testing of the virus, but also to sequence the genome of the virus. And we quickly generated 99 genomes from patients that were coming in with Ebola in Sierra Leone. And we found hundreds of mutations popping up. In just those cases, in just a matter of weeks, this virus was generating all sorts of mutations. And we could take that information, we could start asking some questions. The first thing we were able to show is data that it was known, but it was irrefutable data that the virus was mutating and changing. And we could actually trace back the relationships between those, and we could see that the virus was transmitting from human to human. All of the cases are very closely related, and that really informed the way the public responded. Most of the public health content that we were getting early in the outbreak was about this belief that they were coming in from bats or from monkeys or from people touching fruit that were touched by these different animals. And that was really how we were trying to control the outbreak. But once we started to really see this was human-to-human contact, much of the messaging changed to washing of hands, avoiding contact, tracing individuals who've been in contact with others that were sick. And so genomic sequence data can give you a lot of really key information. But one of the other things that we saw is we started to investigate what are all of these different mutations that are popping up. Most of them don't really matter. Most of our genomes, our human genome, the genome of viruses and other microbes, they don't really have much effect on the virus itself, but every once in a while a mutation might. And we picked up on this one mutation that was in an amino acid change from an alanine to a valine on a protein that is on the surface of the virus. And so there's a glycoprotein that coats the surface of Ebola virus. It allows it to bind to human cells. And what we were seeing is there's a mutation that occurred here. And it was a very intriguing mutation because it was where the human receptor binds as well. And as we did studies, what we found was quite striking, that essentially that one change increased Ebola's ability to infect. And it did so in a way that was humans and primate specific. So it made it less able to infect bats and other mammals and better equipped at infecting human cells. And that essentially one of the things that we were seeing is as Ebola was having more opportunities to transmit from human to human than ever before, it was improving in its ability to do so. And that is why, by the time the outbreak ended, there's at least 30,000 confirmed cases, over 10,000 confirmed deaths. One of the most extraordinary events in our time. And the human toll was, was a catastrophe. And specifically, it affected healthcare workers the most, because healthcare workers are always on the front lines. It's something that we think about a lot here, as all of you are thinking about going to healthcare, is you're on the front lines of these things, and we need to protect. And so Humar Khan, who was there with us in the picture at the beginning, and many of our colleagues at Kenema did not make it past this outbreak. It was, a massive tragedy, and it actually could have been even worse. So this is a picture of Dr. Christian Happy. This is a colleague of mine in Nigeria, who was also the first to do the first diagnosis and detection in Nigeria, in Lagos, a city of 20 million. And here we are sitting at Redemption Church, which is about 100 yards away from Christian's lab, where he detected the first case of Ebola in Lagos, Nigeria, a city of 20 million. And this is basically us sitting on the steps of this church. Just a week after he did that diagnosis, this is what the scene at that church. This is a church that seats 1 million. You can see it on Google Maps. It spans, just acres and acres of lawn. Now 3 million people can sit in this church. At that time, it was right before the annual First Friday, where tens of millions of people were going to descend on this campus where the Ebola was detected. And I often say, imagine if one case of Ebola moved through this crowd. One case of Ebola moved through the crowd we're in today. This is a massive group. I mean, what could happen? And so it just tells us how close we were. We were on the precipice of a cataclysm, and we somehow made it through. But next time, if we're not more careful, we might not. And so my group thinks a lot about what we need to do, right? We need to have better ways of detecting outbreaks in various ways, of connecting information, of cases that are emerging across a grid, of engaging each and every individual to sort of participate, to let themselves be known if they are getting sick, and then to empower every individual in the system. So I'll just tell you a little bit about how we think about that. So as far as detecting, This is the team in my lab at the Broad Institute of MIT and Harvard that were able to do the sequencing of these 99 genomes. We made it publicly available to the web. We shared it with the world. And we worked around the clock sequencing using advanced, massively parallel sequencing technologies to generate these genomes. And the exciting thing about these new technologies is we didn't have to even know that we were looking for Ebola. We can just take a patient sample, clinical sample from an individual, and just generate all of the sequences that are in their sample and find out what is in there. And that's what we did. And we used a standard technique that we used for other viruses, and we're able to generate that. And we need to be able to do that not just in the United States. In that case, we sent the samples over, but that took time. We actually want to do it on ground. And this is actually an image, a rendering of a genome center that's going to be launched. It's actually near completion in Nigeria, made by Mass Design, an incredible architecture firm, using condensed soil built in the backdrop of where it needs to work. We need to be able to do this kind of process in every single country, in every major city. But the thing is, actually, we don't need to have these advanced technologies to be able to do, very sophisticated analysis. For most viruses that might, form outbreaks, we know about them. If you think about it, Zika was first discovered in 1947, Ebola in 1976. Most of the viruses that are deadly threats to humans that may form outbreaks, we already know about them. So we could actually already put in place the ability to detect them right now. And the way we did that during the Ebola outbreak is just simple PCR. You design primers that target a part of the genome, then you amplify that, you generate it, and you read it out on a machine that requires some advanced equipment, but you could do it in a home or in a small lab. And this is essentially what we did. This is an image of our lab in Sierra Leone, and that is the picture of the first confirmed case in Sierra Leone that Augustine Goba here in this picture used. But there's even cooler technologies. You may have heard about a new technology called CRISPR that's revolutionizing molecular biology and has implications on human health. But CRISPR, the system that that's so exciting, was originally designed in nature, originally formed in nature as the bacterial immune system to viruses. It's the way that bacteria can pick up if a virus is circulating. And what happens is there are these guides that the bacteria have, these CRISPR guides, that will circulate around and identify if a virus genome sequence is there. And when it does, it'll call in a nuclease, a Cas protein, and that will cause a cut to happen. And there's a particular type of Cas that is able to do so in a way where as soon as that cut happens, it begins to cut other things. And we can bind that cut to a fluorescent probe to allow you to signal if a virus is in a sample, it'll light up. And that is such a powerful technology that we can actually move this to paper-based. So we have paper-based diagnostics for Ebola and Lassa and other things that are already, we've already implemented in Nigeria and in Sierra Leone and other places to pick up these viruses. And so we really are able to develop technologies at every level, national genome centers, regional hospital clinical tests, as well as paper-based tests that we could almost do at home, available for this. And what we need is now, if we have those at varying levels, we then need to connect that. So during the Ebola outbreak, this is essentially how we were sharing information, on paper, sometimes in Word documents, but a lot of times just with individuals driving around on mopeds trying to share information. And that's just really not going to work. It's quite remarkable how slow our platforms were during that Ebola outbreak and continue to be as the new Ebola outbreak is raging in the DRC. And so what we want to be able to do is connect that. So we have cloud-based technologies that are sharing the information that we're generating at each of these different levels. We have them in Nigeria at our genome sequencing center, at these regional hospitals and village clinics. We also have them in multiple countries in Senegal, Sierra Leone, and Liberia, but this is the kind of network that we want to be creating. The next thing we want to do is engage. So if we have that, so we have this ability to detect in these hospitals and clinics, but we need people to come and to show themselves. And one of the biggest things we saw in the outbreak is it took a long time for people to let it be known that they were sick. And one of the insights I got about this is being a professor at Harvard. I'm on Harvard's Outbreak Response Committee. And a couple years ago, our committee sort of got alerted that there was a number of mumps cases that were showing up in the sports teams. So a lot of different sports teams were having a number of people coming down with mumps. Then we saw it showing up, in non-sports members. We saw it actually in the freshman dining hall, where a number of staff and faculty became sick. in the university health services where nurses had become sick. And then there was a commencement event in which staff of the commencement became sick and then ended up seeding an outbreak in East Boston in a local community. And so we were watching this move across the campus in this way, and we realized that universities are incredibly at risk. Right? And this is well known because you're in close contact with each other, eating together and beyond. And if you think about it, Boston is a laboratory for outbreaks to happen, but it's also a laboratory to think about how to respond to outbreaks, because we were able to sequence samples from all of these different cases and start seeing how different events that were shared across universities were also allowing that outbreak to spread across the city. And so one of the things that we're aiming to do is we've created an app where we've been workshopping it and prototyping it, pairing it with those diagnostic tests to allow people to start reporting in when they're sick. And somebody will say, oh, you have these symptoms, 6 people in your intro to genetics class are reporting these same symptoms, and 2 of them have mumps. And we can move much, much quicker to be able to give you the information you need when you're sick, but also allow that to help other people diagnose themselves and help create sentinel sites for outbreaks. Then we want to empower each individual in the system. So this is a training program we have. We've trained 76 individuals from 6 different countries that have spent an entire summer with us in Boston doing training in molecular technologies and advanced sequencing. We have a program that we've worked with a middle school, the Sarasota Military Prep Academy in Sarasota, Florida. Anybody from Florida here? Yeah, all right. I'm a native, I grew up in Florida, and so I love going back here and working with this middle school in Sarasota each year. They have an amazing simulation that they've been doing of an outbreak where they have clinical teams and they're seeing an outbreak spread. They used to do, they were trying to figure out, well, how do we spread this virus better? Because they're using stickers and things like that. So we created an app that spreads the virus on your smartphone, and we can model every aspect of an outbreak spreading, and we can watch that happen. And essentially these students have to figure out what the virus is, respond to it quickly before they die. And a lot of times, last year 51% of the people died before they figured out what it was. But it's an amazing way in a game situation to find how it feels to be an outbreak when you're not really at risk. And then the final thing is what we're trying to do is we're trying to get every single person to work together, because outbreaks are one place in which everyone fundamentally is working together while we buy time to come up with cures. And the great thing is the genome sequence still tells you so much because the antibodies that we develop are based on the genome sequence, or the proteins that are based on the genome sequence, originally the vaccines as well. And we actually can use that same CRISPR system that I was talking about before that targets the virus to create therapies. And that's another thing that my lab is doing now. What we want to do is create a system by which if we identify a virus circulating, we can, within days, develop diagnostics, within weeks, develop therapies. We need to move that quickly, and we need to all work together to get there as fast as possible and ultimately develop those. So fundamentally, again, it's just figuring out ways of detecting these viruses where they circulate as fast as they're circulating, connect that information across a grid so we have the best information in real time, engage everybody to participate, to work together, to empower every actor in the system, and to work to overcome. And the last thing I'll sort of talk about is, just touch upon is this idea that we have to work together in outbreaks, that ultimately that is the only way that we succeed. It's sort of a war that we're raging, but a war that's won by cooperation. And one of the stories that I often tell about this is that in the villages and in the communities that we work in, one of the things that everyone does before they go into the lab is they get together and sing. And I've always got together and I've sung with the individuals in the lab before we go in and before we do our hard work. And it's amazing. They're dealing with things like Lassa and Ebola, and they start each day singing together. And it's telling us that that is our shared humanity, is what will protect us if there isn't a major outbreak. And right around in the midst of the outbreak, I had been running one of these summer programs where we had 20 individuals that were about to go on the front lines of the Ebola outbreak. And they were training to prepare themselves to go back to Nigeria and Sierra Leone and Senegal and to work at the front of this outbreak. And just every week, one thing that we would do is we would get together and we would sing over this summer, in the middle of the Ebola outbreak. We thought to ourselves, why are we doing this? It's crazy, but we just reminded ourselves, no matter what happens, we just get together and sing. And one of the weeks that we were doing this was a week that we learned that Dr. Khan had become sick with Ebola. Some of the nurses had become sick with Ebola. Within that week, they all had died. And in that same week, Christian Happy diagnosed the first case of Ebola in Lagos, Nigeria. We were in the midst of tragedy, on the precipice of a cataclysm, and we didn't know what to do, but we still somehow got together to sing. And in the middle of it, basically, we were singing, and it was like nobody wanted to be there. Everyone was frightened. But somehow this song burst out of me in the middle of it. And we, as shocking as it is, we just went in and we recorded the song a day before they were going back to the front lines. And we ran around the city right before they went to the front lines, and we made a video. And it's, again, something we needed to do. And the words of the song essentially say, it's a lifetime for one proof, that I'm alive and so are you, and we're here and we are the proof. And fundamentally, we kept singing about this idea that we strive and we work and our hunger won't die, and we're in this fight together. So look at I'm gonna just play you a quick clip of these individuals right before they went to the front lines of the Ebola outbreak. Thank you. All right.
Dr. Sabeti, I am so excited to be here with you. Most of you guys would not know this, but I was actually in Dr. Sabeti's class that she teaches at Harvard. So she is actually my professor, and I was as awestruck by her the first time I was in her class as I still am today. So thank you for joining us. Thank you for being here and sharing some of your story. We have a lot of questions from the audience, and they range from how it felt right after seeing the sort of impact you were able to have. But the one I wanted to start with is actually about the challenges that you faced in beginning. So what sort of issues did you have in a career that was starting almost back in high school? What sort of advice would you give to these students who might be facing those challenges now?
Yeah, a lot of things. There's a lot to go to. I mean, I think at the end of the day, I'll give you a few. Solidarity is really important. Nobody will go through their career without being beaten down at some point. At the end of the day, human nature is crazy, and there's a lot of different challenges that you'll have over and over again. And ultimately, you need to find individuals who you can navigate it with. And I have gone through unbelievable challenges in my career, and most people, on Instagram it looks like our lives are great and everything's wonderful, but there's a million challenges from here to there. But I think ultimately finding a good group of people that can help you with it is important. And a big thing I often say is also putting your own health first. I'm an MD, but I'm pretty hard on the medical community because ultimately, if you look at most of our doctors, they're not that healthy themselves. And how can you actually try to promote medicine when you yourself are not healthy? You're not sleeping, you're anxious, you're depressed, you're not exercising. And ultimately, I know everybody in this room, to be where you are, have the potential to be anything you want in the world, and we've heard a lot about that, and I firmly believe that. But the one thing that can stop you from getting there is your health. Be it physical or mental, it's your ability, because challenges will come and you have to be ready for anything and you have to put yourself first. It's the most important thing. Medicine does have, incredibly high rates of depression and things like that because it's a hard job, but you have to do it with joy and you have to put yourself first. And so I think that's the biggest piece of advice I can tell you is go for walks. There's nothing that an extra 10 minutes of sleep, 10 minutes of work isn't going to help. Sleep will be more important. Ultimately, exercising, feeling good is more important to your long-term success. And I would not be where I am today if I didn't do that.
Absolutely. And I think what you said before about surrounding yourself with people who can encourage that sort of thing is also incredibly important. To change gears a little bit, something that I've always found fascinating about your work is the fact that you work in a very global context. And I was wondering if you could speak a little bit to the challenges of kind of going to different cultures and sort of any problems or any advice that you would have for people who kind of want to translate the research done in Florida to somewhere completely different in the world.
Yeah, I mean, I think a big thing about that is just to listen, to basically there's so much you can learn from different cultures. It's very, it's a lot of times people think, oh, and I'm going to go to Nigeria and I'm going to help them, or I'm going, and it's kind of laughable because they're so sophisticated. The doctors are amazing. A lot of them are much better at diagnosing cases than we are because they have 80 times the workload that we have. And so I think a big thing is just when you go, look, learn, and contribute in small ways. A lot of times a danger like everyone has is they feel as if by high school you should have cured cancer. And the people who are the most successful in like the long term are the people who just keep learning their whole lives. You don't have to feel like you've accomplished anything at any point. Just keep learning, keep listening, keep watching. Go help in small ways first, and then in big ways later. I think that's like what I believe is the best way to go, is to really go as partners and to go to learn.
Yeah, I think that's great advice, and it's definitely a much more humble way of approaching a context that you don't necessarily know that much about. You mentioned in your talk a little bit about CRISPR and some new technologies that are coming out that are really promising, but also come with their own set of concerns and things that can happen. And I was wondering if you could speak a little bit to the responsibility you feel as a scientist for creating sort of ethical guidelines around these new technologies.
I mean, I think that ultimately it is very important that scientists participate in the discussion around how these things go. And I often get asked this question of, are we training too many scientists because there aren't that many faculty positions, and so should we just train less scientists? And my mind, when I talk about that, I say, absolutely not. We should train more. That ultimately, whatever job you go into, if you go into medicine, if you go into science, if you step out and you do something completely different, using a scientific framework as which you come at the world is really important, because you'll have a hypothesis, you'll think about what controls you need to do to test it, how to prove it, how to go out in the world. We need scientific thinking in everything, including the policy. We need to think about what does it mean. We need people that have that background to engage and to participate. And if we step out, it's very dangerous because you'll have a lot of politicians who know nothing about what they're discussing, invoking policies that really matter. And so, I think that there's a lot of, in the scientific world, there's a lot of pressure to just kind of keep your head down and just keep moving the science forward. But I think that that's actually negligent not to participate, to engage on a larger scale. So everything I do within the outbreak world, I kind of write a policy piece that goes along with it. I'm constantly thinking about thinking in every direction where this could go. And any of these technologies we create could change the world. Hopefully for the better, but sometimes for the worse if we're not thinking about it and engaging with it.
I think that's a great point, and you've definitely taken a very active role in training the people who will be creating these policies. I was actually wondering, how do you translate your work in the field and in the lab to your teaching that you do in the classroom?
Translate the work I do in the field and the lab. I mean, I think if you took the class the first year I taught it, it's gotten more and more ridiculous. Was there a t-shirt cannon when you were in my class?
There was not.
Okay, yeah, I got one of those later. Yeah, I mean, so the class I teach has 500 students. It's like a, intro to genetics, like a class that most bio majors have to take at Harvard. And I just have fun. I mean, I have fun with everything I do. So I throw clementines to get people to answer questions. I pump in music. It's a lot like this, actually, but not at this scale. But I have a great time. I think that in my mind, there's no reason why you can't be learning about really complex facts in genetics and not be enjoying each other. And so to me, every part of what I do involves like being very, very serious about what you're doing, but then having joy in when you're doing it. And so I don't know whether or not that's like a learn from the field, but it's just enjoy people, enjoy the people around, and have a good time with it. So yeah, I'd love you to come back and see the class now. I've had
I remember the Clementine throwing.
Okay, yeah, there you go. That one I
was
got.
always scared I would miss it. You are absolutely a pioneer in what you do. So we actually had a really great question from Grace in the audience who wanted to get your thoughts on what you think the next big breakthrough is going to be in immunology.
In immunology? Well, I mean, I think in general, like with all these fields, the possibilities of engineering systems is quite extraordinary, and we're getting better and better at modeling what nature does. So a lot of the types of things that we've done in the past has been to develop, drugs that we found that we've tested in this very we've just discovered them by chance, but now we're really understanding the human genome, how it works, the human immune system, how it works, and modeling it and engineering it. And so a lot of these antibodies, these sort of polyclonal antibodies that we're creating, these that are tracking what the human immune system does and improving upon it, is going to be really fascinating. And so I think in general, we're understanding better how the human immune system works and modeling it in cancer therapies and beyond. And then we're doing the same kinds of things in our case, seeing how the bacterial immune system works, or there's some things that we are actually learning from the viral immune system. We're trying to take everything that nature's already figured out for us and then modeling it and then improving upon it. And I think that that's going to be a really exciting time. The more we mine the diversity that is in nature to understand the best approaches to use.
That's fascinating, and I think the coming years will be really interesting to see how some of the people in our audience kind of shape this field as well. I could talk to you forever, but I think I'm out of time. But thank you for sharing your insights and your thoughts, and for sharing your joy for everything you do.
Well, thank you, Shri. It's amazing to see you here. Thank you, guys.